Hello dear students, today we are going to do the anatomy of flowering plants. So first of all we have to understand the tissue system. Tissue system is a group of cells and on the basis of location and function there are three type of tissues which are found in the plants. Epidermal tissue system, ground or fundamental tissue system, vascular conducting tissue system. So first of all we will do epidermal tissue system. So it forms the outermost covering of the whole plant like skin. So epidermal layer is there for the formation of the body and outer part of the body, and it is made up of epidermal cells, stomata and epidermal appendages. When we talk about the epidermal tissue system, so epidermal layer forms the outermost layer just like this. This is a epidermal tissue system in which we can see that this is a layer of single cells, right? And this is the epidermal cells. So we can say a small amount of cytoplasm is present in this and present inside the cell wall. This is the stomatal pore, and these are the guard cells which control the opening and closure of the stomata. This is the barrel shaped cells called as bulliform cells, which generally absorb water and make the leaf in expanded form. This is the epidermal appendages, yani the hair like means hair like projections which are present on the stem. Now we can say कि just in the neighboring of guard cells there are subsidiary cells which control the opening and closure of guard cells. So this is the basic structure of epidermal tissue system. So next we can see that epidermis is made up of elongated, compactly packed or arranged and form a layer, usually single layer, usually made up of parenchyma cells and having the vacuole to store the food. Epidermis protect the plant body and small amount of cytoplasm lining the cells are present and have large vacuole to store the food. Thick waxy layer is present above the epidermis that is called as cuticle. Cuticle prevents water loss, that is the transpiration. Cuticle जो है that is absent in the root. And now we will do the epidermal appendages, that is the outgrowth present on the epidermis. So if this outgrowth is present on the root, it is called root hair, and if this outgrowth is present on the stem or shoot, it is called trichome. So the function of root hair is to absorb the water and to increase the surface area, and trichome prevents the water loss. Now the stomata, the two main function of stomata are there. They help in the gases exchange and help in transpiration also. Gases exchange means O2 and CO2 exchange from the environment. Now this is the structure of stomata. In the dicots, there is a different type of structure found in the dicots and monocots. So in dicot the structure is just like this. You can see this is the stomatal pore which is guarded by two kidney shaped cells called as guard cells, and guard cells usually have these chloroplast. Guard cells have chloroplast. And now the around these guard cells some special type of cells are also found which is called subsidiary cells. What is the function of subsidiary cells? Provide the mechanical support to the guard cell and help the guard cell opening and closure, right? So this is the bean shaped cell. This is the epidermis which is the outermost covering. Now the inner layer of guard cell, जो inner layer of guard cell है that is thick and the outer layer of guard cell is thin. This is the bean shaped guard cells present in the dicots. Now the monocots, the structure of stomata is dumbbell shape. These are like just like a dumbbell that are used for exercise. This is the stomatal opening. This is the guard cells which are present. This is the subsidiary cells, and this is the epidermis. This is the epidermis. So we can understand that monocots and dicots have different type of structure of stomata, different type of structure of stomata. Now stomatal apparatus. Stomatal pore, guard cells and subsidiary cells together called as stomatal apparatus, right? So these three structures are making together stomatal apparatus. Now epidermal appendages. Appendages means outgrowth which arise from any part of the plant, right? So number of hairs or appendages are found on the cells of epidermis. They can be of two type, like earlier I explained ki root hairs and trichomes. If they are present on the root, then they are called root hairs, and if they are present on the stem, they are called as trichome. The function of root hair is to increase the surface area so that maximum absorption of water and mineral should be there from the soil, and trichome's function is to just close the stomata or lenticels present on the stem and to prevent the water loss, right? Now the second part of the plant is ground tissue or fundamental tissue, which just make the body of the plant, and all the tissues except the epidermal that we have right now studied. All the tissues except epidermal and vascular bundle are combined to form the ground tissue. So generally it consists of simple tissues like simple permanent tissues like parenchyma, collenchyma and sclerenchyma. Parenchyma cells are generally present in the cortex region, that is the outer part of the plant. Pericycle, pith and medullary rays in the primary stem and root. Now let us first know about these terms. Ki what are these terms? Cortex is the outer layer of stem or root present below the epidermis. Just after the epidermis, the structure which is present is called as cortex, but outside of vascular bundle. It is outside from vascular bundle and inside from epidermis. Pericycle, a layer of parenchyma between the endodermis and phloem, is called as pericycle. So these structure are important to know. Now pith is present in the center of the stem. Now medullary rays, you can see this is the pith which is present in the center of rays, and from this pith the lines arise which are called as medullary rays or pith rays. These lines are called medullary rays or pith rays generally, or vascular rays. Medullary rays are the strips of parenchyma. Basically these strips are made up of parenchyma tissue, which is a living tissue. Now the leaf, this is the structure of leaf, and the structure of leaf have, this is the outermost layer that is called as epidermis, and below the epidermis you can see the cells which are present, which are thin walled cells. These cells are called as mesophyll cells, and they are chloroplast bearing cells which are found below the epidermis, and if the chloroplast is there, then generally the chlorophyll is present and do the photosynthesis. Now the vascular tissue system or conducting tissue system. These are made up of complex tissues, that is xylem and phloem, and they together makes a vascular bundle. Now vascular tissues in the stems. First of all we will see the types of vascular tissues. Now this one structure we are going to know. This is the xylem and this is phloem, which are present alternatively. You can see one xylem, one phloem, xylem, phloem, xylem, phloem, xylem and phloem. It means that xylem and phloem are not together present, they are separately present on the different radii. Xylem and phloem is present on the different radii as well as alternatively. So such kind of arrangement of xylem and phloem is called as radial vascular bundle. So generally radial vascular bundle is found in the roots. They are present in the roots. Next is the type of vascular bundle which is found in the monocote and dicote. Here you can say the xylem and phloem and xylem is present on the same radius. This is phloem, this is xylem, right? So this is called as conjoint vascular bundle, but it is of closed type because though no meristematic tissue is present between phloem and xylem. Generally this is found in the monocots. Next is conjoint vascular bundle but open type. Why it is called as open type? Because we can see there is, this is phloem, this is xylem, and this is a strip is present between xylem and phloem. This strip is made up of meristematic tissues, which are the continuous dividing tissues and called cambium. Generally this is called as cambium, which is present in phloem and xylem. It means ki if the need of the growth will be there, then this cambium will start forming the secondary xylem and phloem. So this is conjoint vascular bundle but open type. This is found in the dicot. This is now the explanation part of this one. In dicot stem, generally cambium is present between xylem and phloem, right? And the type of arrangement is called as conjoint open vascular bundle. Because of presence of cambium, or which are made up of meristematic tissues, these vascular bundles have the ability to form the xylem and phloem whenever the needed, right? So basically they are responsible for the secondary growth of the plants of dicots. Open type of vascular bundle. Monocot stems, xylem and phloem don't have cambium between them and don't have secondary xylem and secondary phloem, and they don't do the secondary growth of the plant, so called as conjoint closed vascular tissue system. Example है bamboo and grass, which are generally monocots. Now radial vascular bundle, that we have already seen, this is found in the roots, and generally xylem and phloem present in the alternate form or alternate manner along the different radii, and generally they are present in the roots of plants. Now we will understand this with the help of this diagram. Now this is the outermost layer. Actually this is the TS of dicot root. TS means transverse section. TS means transverse section. This is the structure which is called as epidermis, and over epidermis is hair like projections called as root hair, and epidermis is also called as epiblema. It is also called epiblema. Just below the epidermis there is number of layer of cells called as cortex. Now under these cortex some cells are elongated cells are found that are called as endodermis. As they are endodermis, so they are present inside, so they are called as endodermis. Now you can see this pink color layer of cells. This is called pericycle. This is called what? Pericycle. Now you can say this type of cells which are present here. So this is xylem. So radial vascular bundles, these are radial. This group of cell is phloem and this is xylem. This is phloem, this is xylem. This is phloem, this is xylem. This is phloem and this is xylem. So we can see that radial vascular bundles are present, xylem and phloem present alternatively, right? Now I hope, and in the center there is pith, which is the central part of root. Pith is forming the central part of root. Now I am going to explain the dicot root. First of all, anatomy of dicotyledonous and monocotyledonous plants. So when we cut the transverse section, right, so we can see such kind of structure which labeling I have explained. So epidermis is outermost single layer. You can see this is the outermost single layer, which is also called as uniseriate or unicellular. It is, this epidermis is also called epiblema, right? It is also called as epiblema, and the cells of epidermis protrude outward and they just form the root hairs, right? So hypodermis, which is present below the epidermis, is absent in the roots, and cortex consists of several layer of thin walled. Now you can say that cortex region has number of layer of cells. Cortex region has number of layer of cells which are made up of parenchyma with intercellular space present between them. Now you can see they are not tightly packed. They are not tightly packed. Some intercellular space is present between the cells, right? So intercellular space is present between the cells. Epidermis, endodermis, this is the endodermis, which you can see, this is the endodermis, this before the this pink line. Endodermis most layer of cortex, and the cells of endodermis are barrel shape. Now you can see these cells are barrel shaped cells. The cells of epidermis are barrel shapes and they have Casparian strips or Casparian bands. They have Casparian strips or Casparian bands present on them that I will show you the diagrammatically. Now you can see the Casparian strip. These are the endodermis cells, and which I am trying to show you that the structure. These are barrel shaped cells in which you can see the Casparian strips. This is the Casparian strips, which generally act like a checkpoint for the entry of unwanted material in the plants. Casparian strips act like a checkpoint. It will not allow the unwanted material to enter along with the water. These Casparian strips are made up of suberin, and this is the plasma membrane, this is cell wall, this is Casparian strip, and the Casparian strip is made up of suberin, and they will act like a checkpoint. This is the pericycle, and inside the pericycle xylem and phloem bundles are. The next part will be explained in the next video.